Hello, welcome to Progressive Education Society's Modern College of Pharmacy, Nigdi, Pune, in the e-lecture series of in the subject Natural Drug Technology.
Hello, myself Dr. Bhushan Pimple. In this video, we will discuss about liposomes and phytosomes as per the syllabus of Savitribai Phule Pune University. Here, we are going to discuss the brief overview about these formulations. Liposomes, these are vesicular systems and they contain the biodegradable polymers. The liposomes themselves are colloidal in nature and their shape is spherical. The bilayer that is contains a hydrophilic head and a lipophilic tail. Usual size range is from they can exist as a nanoparticle or even as a microspheres. So the size range varies from 20 nanometer to 3 or 3.5 micrometers in diameter. The bilayer, it entraps an aqueous core. Now here the orange color portion highlights the aqueous core. So if you want to entrap an aqueous drug or a hydrophilic drug, this can be entrapped at the centermost zone or the core region. Simultaneously, it also allows a lipophilic drug to be entrapped into the lipid bilayer. Now, this black colored zone indicates the lipophilic region. So, lipophilic drugs can also be entrapped here. Furthermore, amphoteric drugs, the drugs that are slightly lipophilic and slightly hydrophilic, can also be carried by these vesicles. The hydrophilic part gets oriented towards the aqueous core, whereas lipophilic part of the drug gets oriented towards the lipophilic zone. The bilayer is made up of lipids, and these lipids are having the hydrophilic head and lipophilic tail. Now this bilayer, the bilayer can be prepared from naturally derived lipids or sometimes synthetically prepared lipids in the lab, or semi-synthetic lipids can be obtained, and those can be utilized for the preparation of these bilayers. This is the alignment of the bilayer. The head is made up of hydrophilic side. So you can notice here the orange color, suppose the orange color zone is water, then the head of the inner core is getting aligned towards the water. Whereas the tail being lipophilic, that means the lipid loving, it is not getting oriented towards the water region. Instead, it has formed a sandwich like layer at the periphery. When the lipids interact with water, the polar lipids self-assemble and form self-organized colloidal particle. That means in a beaker, if you add water and lipids and sonicate them, the lipids they themselves align in the manner as depicted in the picture. That means they are self-assembling. Hydrophilic compounds can be entrapped in the water zone. Lipophilic compounds can be entrapped into the lipids. Now, this is the zoomed image, or you can say animated image of liposome. And Cipla has designed amphotericin B liposomes. So you can see here the amphotericin B drug is entrapped into the lipophilic zone because amphotericin B has dual nature. It has some molecules of lipophilic nature and some of hydrophilic nature. So the molecule of amphotericin B has aligned itself in this manner. The method of preparation, usually it involves four basic steps. First is drying the lipids from an organic solvent. That means the lipid is dissolved in an organic solvents like chloroform or ethyl acetate, benzene or solvent ether. And the solvent is slowly evaporated, dispersing the lipids in an aqueous media. The solvents along with the lipids are slowly injected into the aqueous phase, and the mixture is heated. Upon heating, the solvent goes off, that is solvent ether or chloroform, it goes off and it leaves behind the lipids. The lipids that are left into the aqueous zone or aqueous area, they align themselves to form these kind of liposomes. Further, these liposomes, not all of these lipids they are able to align in a proper way. So these liposomes need to be purified and finally should be analyzed for their particle size or shape or drug entrapment efficacy, etc. Advantages. Since we are discussing only the brief overview about the novel drug delivery systems, we are not going into the details as to how the liposomes are prepared and how the herbal molecule is entrapped into it. That we are not going to discuss in this video. The advantages of liposomes are that liposomes can allow one or they can have one or many such layers of lipids in order to form multi-layered ball or a sphere. This helps to accommodate the many molecules of lipophilic drugs. Liposomes have more solubility in water compared to the normal lipophilic polymer. And since because our blood is made up of more amount of water, the bioavailability of the drug, if injected or administered through liposomes, it increases drastically, thereby reducing the dose of the drug, thus making the drug more potent. In vitro and in vivo stability is also excellent as far as liposomes are concerned because they are more stable in this form. The drug is more stable in this formulation because of its encapsulation. Further, the drug is not directly exposed either to the external atmosphere or even to the internal bioatmosphere like pH or tissue fluids or certain other toxic drugs which are co-administered or other drugs which can be co-administered. So this also helps in reducing the drug-drug interaction. Liposomes can encapsulate hydrophilic drugs in the core region, lipophilic drugs in the membrane, and amphoteric on the interface of lipid and water. They provide a proper flexibility to couple with site-specific ligands to achieve target drug delivery system. That means if the liposomes are coupled with certain ligands are um, molecules that have specific orientation which is complementary to the receptor. So suppose if you want to target to the cancerous tumors. So cancerous tumors, we need to know the exact shape of the receptors of a cancerous tumors. In that case, you can make or attach ligands which are having a complementary shape as that of the receptor. So the liposomes will be carried specifically to the cancerous tumors. Since they are non-toxic and provide flexibility of entrapment of the drug molecule. Also, the polymers incorporated into formulation of liposomes are biodegradable, completely biodegradable. They are non-immunogenic. They do not initiate antigen antibody reaction, or these are not identified as an antigen by the body, so can be directly injected into the systemic circuit. A few more advantages of liposomes are they act as a carrier for small cytotoxic, that is anti-cancer molecules, and acts as a vehicle for even large molecules like gene or protein. They can produce sustain and control release as well, depending upon the polymer that we are making use of. Further, they can change the PK profile, and not only of the drugs but herbal drugs and vitamins or enzymes, those are administered through the liposomes. However, liposomes also have certain limitations like their solubility. The solubility, because of their lipophilic profile or lipophilic coat, sometimes is less. Also, the drugs cannot be entrapped. All the drugs cannot be entrapped and into the liposomes because of their poor solubility or pH dependent solubility. They have shorter half-life compared to a sustained drug delivery system. Sometimes phospholipids undergo oxidation. As we all know, lipids have a tendency to undergo photooxidation. So there are chances that that may result into certain metabolite formation, which can cause adverse reactions or even hydrolysis of the phospholipids. The liposomes administered can be punctured or can ooze out the drug at unwanted site. This has been recorded that there are certain drugs, or there were certain cases wherein the drug used to be leaked at before the target site. So leakage and fusion of many of the encapsulated drug can occur. And lastly, the production. Yes, liposome production is much more costlier than the traditional formulation. That is the reason why the entire final product has some higher cost. Examples of herbal liposomes are like entrapment of the drugs like quercetin, flavonoid isolated from various drugs, herbal drugs, then silymarin extract isolated from Silybum marianum and capsaicin. The excipients used in these formulations were egg phosphatidylcholine and PEG. For silymarin extract, the excipients were cholesterol and sterylamine, and same was the excipient for capsaicin. The applications of these formulations like quercetin liposomes were used to increase the cognition and anxiolytic for its anxiolytic effect. Further, the bioavailability of quercetin is very poor in its intact form, so or in its natural form. So, on addition or conversion into liposomes increases its bioavailability and decreases the adverse effects. Silymarin is basically used as a hepatoprotective drug, and its hepatoprotective activity has been and the efficacy has been increased, has been found to be increased. Capsaicin is used as a permeation enhancer for other drugs. So you can find most of the arthritic or anti-arthritic drugs coupled with capsaicin. So capsaicin increased the permeation and also increased the activity when administered through. So these are few of the marketed liposomes like liposomal iron capsules or curcumin and certain night cream containing liposomes and vitamin C. Yes, you can find range of different marketed products containing vitamin C as the major ingredient for liposome formulations. Phytosome. Next formulation is phytosome, also known as pharmacosomes. These are especially designed for accommodation of herbal drugs. They consist mainly a drug and phospholipid complex and have herbal active ingredient, especially like suppose isolated components like curcumin, quercetin, and all. Rarely they do have extracts as well. The principle for formulation of these phytosomes is they are these phytosomes are complexed with or the drug is complexed with the phospholipids which are used in development of phytosomes just to increase the bioavailability of the herbal drugs. When water soluble drugs like herbal drugs, if they are converted into phytosomes, they form a compatible lipid complex. This ensures their stability as well as PK profile. Definition: it is an association of a herbal drug and lipids. The lipids and herbal drug they form a complex and that are developed using stoichiometric ratio. Stoichiometric ratio in sense a proportionate ratio or 1 as to 1, 1 as to 3, 3 as to 1, whatever the case is, the ratio is constant of lipids and the herbal constituents. And both of these, that is lipids and the herbal constituents, are dissolved in the same solvent and the solvent is slowly evaporated. So the lipids they come closer to the herbal drugs and they entrap or form a bond with the herbal drugs. So this is how the phytosomes are prepared. The interaction of the drug and the polymers of the phytosomes is the functional or the polar functional group, that is OH and H of the herbal drug interacts with the phosphate group or ammonium group of the polar head of phospholipids by simple HH bond or hydrogen bonds. Like here, we have shown the two different vesicular formulations. Now, this particular green colored formulation containing black star shaped drug is a complex of the phosphatidylcholine or phospholipids and the drug. Now, this is how a phytosome appears. Whereas if you just compare it with the liposome, in case of liposome, the upper half in this image, the upper half designates a liposome. In liposome, the herbal drugs usually being polar are entrapped only in the aqueous core. Now you can see here the star or black stars are present only in the aqueous core. That means herbal drugs can also be incorporated into liposomes, but they are generally prefer to remain into the aqueous core. But as far as phytosomes are concerned, the herbal drugs they form a complex with the polar head of the phytosome. So here the herbal drugs are getting entrapped into the polar heads. So this is the basic difference between a liposome and a phytosome. Therefore, the phytoconstituents, generally the herbal phytoconstituents, they get properly attached with the phospholipid polar head. Example of the phospholipid polar heads are phosphatidylcholine and a complex with the herbal is of catechin. H bond is formed, hydrogen bonds are formed between the OH group of the phenols. Phenols in case of catechin. Catechin is a polyphenolic compound. So OH present in the phenol of the catechin interacts with the phosphate group present in the phosphatidylcholine, and this interaction is weak HH bond interaction. The phytosomes, they do have certain limitations like being water soluble. All the phytosomes, they have more amount of polar heads, so all of them, they are properly water soluble, so they are poorly absorbed through the gut or intestine. So most of the phytosomes, they get flushed out. They have poor membrane permeability. In order to permeate any membrane, lipid membrane, the molecule or the drug should have slight lipophilicity. So both the herbal drugs as well as the phosphatidylcholine have very weak lipophilicity. So the membrane permeability is a challenge. And because of this, they have a poor bioavailability. Most of the drugs, when taken orally, are degraded. Most of the herbal drugs are degraded at GIP. These are the few of the limitations. Now we'll talk about the advantages of phytosomes. When you are complexing the herbal drug, the bioavailability is increased. Here we can see a controversial statement. Increase in the bioavailability and absorption is more compared to non-complexed one. That means if you are administering drug alone, herbal drug alone, let us say suppose a curcumin powder which is added into a capsule and given orally, plain capsule and orally, given orally, and if you're comparing this formulation with the phytosome, in that case, bioavailability of phytosome is more compared to a plain curcumin capsule. Since they are slightly lipophilic in nature with a definite melting point, their solubility is also proper in non-polar solvents, and they have moderate solubility in fats. Moderate solubility, not more solubility as compared to liposomes. They increases the stability profile by improving drug absorption to the intestinal tract as compared to unbound phytoconstituent. Here we are in the advantages, we are comparing the phytosomes only with the pure drug or pure phytoconstituents, and forms a stable complex with the phospholipids. Can be used for liver targeting drug as they can increase the solubility in the bile salts. Bile salts, we all know, is are emulsifying agents, so they can immediately take up the moderately soluble lipid soluble drugs or phosphatidylcholine. Decreases the drug dose because of its potent the bioavailability. As the bioavailability is increased, so the drug that is to be loaded on phytosomes is reduced up to 60% or 40%. These are few of the marketed formulations of phytosomes, like generally green tea phytosomes or milk thistle means Silybum marianum, which is used for treatment of liver disorders, and curcumin, grape seed extract, or hawthorn and grape seed phytosomes are available in the market. Compared to liposomes, the cost of phytosomes is much smaller or cheaper.